It's a minute after noon. So I'd like to, a few people are still arriving, but I'd like to welcome everybody today to the Cancer Center Grand Rounds. And for those of you who don't know me, I'm Mark Lemon. I'm stepping in for Eric Weiner today because Eric is otherwise engaged. I'm Mark Lemon. I'm Deputy Director of the Cancer Center, and so I'm channeling Eric actually, which is what I'm doing. So, but great to have you all here, and great to have Luisa Escobar-Hoyos and Rosa Munoz Xicola to talk with us today. And we begin with Dr. Luisa Escobar-Hoyos, who is an assistant professor of therapeutic radiology. She received her master's degree in biomedical sciences at the Universidad del Valle in Cali in Colombia. And then as a Fulbright scholar, she pursued a PhD in cancer, molecular and cellular pharmacology at Stony Brook University, where she was mentored by Dr. Kenneth Schroyer. Then completed her postdoctoral training at Memorial Sloan Kettering Cancer Center, co-mentored by Dr. Stephen Lynch and Omar Abdel Wahab. And the overarching goal of Dr. Escobar-Hoyos' lab is to develop new approaches to tackling pancreatic and lung cancers, with lots of really very exciting and new work going on, and lots of innovation. And specifically, her team is currently seeking to understand and target somatic mutations, and importantly, aberrant RNA processing in these tumors in order to develop novel therapies. So it's a great pleasure to have you start us off, Luisa, and I look forward very much to your talk. So please take it away.
Wonderful. Thank you, Mark. Let me can everyone see my presenter mode? Sorry, not my presenter, my full slide.
Looks like a full slide. That's all very good.
Perfect. So thank you, Mark, again for that nice introduction. And thank you everyone for participating today in the Cancer Center Grand Rounds. And I'm excited to share with you a little bit of the research that we've been doing in our lab in terms of finding how altered RNA splicing is a driver event for pancreatic cancer. So a few disclosures. I'm part of the scientific advisory board of KDX Diagnostics. I won't be presenting the work that I have with them today. I'll be talking about this compound, the small molecule inhibitor of splicing that has been provided to us by H3 Biomedicine. And currently we're in discussions to launch a clinical trial using this compound based on the research that I'm going to show you today. So for many of you, it's not unheard of that pancreatic cancer is a very lethal malignancy. And in here is just plotting the survival over time for the major cancers. And what we can see in this evidence is that unfortunately, we haven't been able to make that much improvement in the 5-year survival rate of pancreatic cancer. And this could be attributed to many reasons. It's a disease that is diagnosed once it's already systemic. The first line chemotherapy and immunotherapies are ineffective. And the available targeted therapies are only available to 1% of the cases that have actionable mutations. So there is a really strong clinical need to understand more of these tumors and develop new treatments. So just to introduce a little bit of the mutational landscape for these tumors, and mainly today I'm talking about pancreatic ductal adenocarcinomas, or PDACs, the most common form of pancreatic cancer. So we know that they are driven by a first hit mutation, activating mutations in KRAS, and also is very common to find P53 mutations. So we'll talk about a little bit more of these mutations. There's also sometimes it appears that other tumor suppressors are mutated, and then after these 4 top genes, there's really a sea of mutations that appear at a very low frequency. So using mouse models over the last 15, 20 years or so, we have been able to kind of dissect a little bit the genetics of this disease. We know that the normal pancreas, if we engineer KRAS mutations into the acinar cells, these mice will start developing panning or pancreatic intraepithelial lesions that will progress into pancreatic cancer if we add an additional mutation in P53. And so basically this tumor follows kind of a 2-hit hypothesis Knudsen model, and all to enhance the activity of OncoKRAS over the time. Now, for many years, we've known about these two mutations driving this disease, but really we haven't made much effort to understand how these oncoproteins cooperate in the case of pancreatic cancer and even other tumors. So a few years ago, when I was going to start as a postdoc at MSK, these studies came out on the molecular subtypes of pancreatic cancer, with the squamous and basal subtype being the most aggressive molecular subtypes. And when you look into what genes are differentially expressed into this subtype, there is a small subset of genes that it's overexpressed in these molecular subtype. And when you look into their mutation status, they're highly associated with gain of function, mutant P53. And these genes that are being enriched are, the majority are coding for splicing regulatory proteins. So after I read these reports, I kind of got interested on understanding a little bit more alternative RNA splicing. And just to remind everyone, during alternative splicing, not only we remove the introns of genes, but also there could be a selective retention or skipping of exons. And this can lead to proteins that had opposite functions or no protein being formed. And we all have our favorite gene. And sometimes we don't study the alternative splicing of this gene. And this pathway in general is a very potent and plastic that can actually explain a lot of the features that happen in cancer cells. So based on those reports, I asked the question, is there a connection between mutations in P53 and alterations in RNA splicing? And so first we took the RNA sequencing from many patients and we divided them into 3 different groups. Either if they had truncating mutations in P53, meaning loss of function mutations of P53, gain of function mutations, so mutations that make the protein going from a tumor suppressor to an oncoprotein, or wild type P53. And we compared the splicing differences between these tumors. And in the case of pancreatic cancer, the most common hotspot missense mutations are these 4 listed here. So here are the first results. Well, so in the x-axis, you have a measurement of alternative splicing of exons in the different tumors. And in here is each one of the mutations of P53 compared to the wild type P53 tumors, and each one of the hotspot mutations compared to truncated P53. And what you can appreciate is that all these hotspot gain of function mutations change alternative splicing, and the R175, one of the most common ones, actually changes the most. So based on those correlation studies, we started asking, well, is P53 changing splicing in pancreatic cancer? So we went ahead and developed 3 different model systems, patient-derived organoids, where we can actually shut off the expression of the mutant P53. And after doing deep RNA sequencing and splicing analysis, we can see that there are these different exons that are either preferentially retained in red or preferentially spliced out in the context of mutant P53. To complement this model, we generated a murine cell line also with the same capacity to shut down mutant P53. And again, we were seeing these changes or swapping of exons. And lastly, we took pancreatic precancerous mouse panning organoids, where we knock in the mutation of R175. And again, we were seeing that even in early stages, after early knock-in of the mutation, we were seeing this differential splicing of exons. So we wanted to ask, what are the specific features of these exons that are being either retained or skipped? And we found that there is the retention of these exons is not random. All the promoted exons after splicing, those ones that are going to be retained in the mature mRNA, are highly enriched for Cs, while the repressed exons were highly enriched in As and Gs, suggesting that this was pretty much a program established in these cells. So we wanted to focus on what were the mRNAs that were being coded by these gain of poly-C exons. And so here we're zooming in into one of these mRNAs, GAP17, where in mice and in humans, this exon 17, which is a poly-C exon, is alternatively spliced. And in here are the raw sequencing of the reads of this exon. And what you can appreciate is that whenever the mutant P53 is present, there's higher reads, versus when you knock it down, there is a decrease on the retention of these exons, but not the neighboring exons. And we saw this pattern across the murine cell line that we had engineered and the panning organoids that we had also CRISPR'd. So from here, we actually went and said, well, let's go back to patient-derived samples and let's see if the retention of these poly-C exon in GAP17, is it exclusive for the R175 mutation, or is it also found in other gain-of-function mutants of P53? And the answer was yes, it's actually retained in not only R175, but other gain-of-function mutant P53s, as you can appreciate here from this targeted PCR. So then the question was, well, what is the consequence of incorporating poly-C exons into mRNAs over time? And so when we started looking at all of the mRNAs that were incorporating poly-C exons in the presence of mutant P53, we found that a family of proteins called the GTPase activating proteins, or GAPs, were actually gaining these poly-C exon. In fact, 25% of total GAPs encoded by the human genome were gaining poly-C exons. And just to remind everyone what GAPs do, they actually accelerate the GTP hydrolysis of RAS proteins. So they bring it from the on state, which is bound to GTP, to the off state bound to GDP. And just to give you a sense of how was this exon impacting the protein, we were seeing that these poly-C exons were in frame, and when they got translated, they encoded for prolines, highly rich proline tails in the C terminus of the proteins. And here is just an example to show you that actually these are different molecular weights of the protein. Here is the promoted by P53 with the poly-C exon, and here's the repressed P53 isoform of GAP17 without the poly-C exon. So both can be produced and translated in the cell. So at this point, we were faced with the question, well, what happens with the KRAS state of either GTP bound state or GD bound state whenever you have a plus poly-C GAP17 or a minus poly-C GAP17? And so we did this in cell experiments where we actually first overexpress either the poly-C GAP17 or the minus poly-C GAP17. And we actually did a pull down to capture GTP bound KRAS. And then we used an antibody that it's only recognizing the mutant form of KRAS to determine the levels of active KRAS in these cells. And what we found was interesting, which is in the presence of poly-C GAP17, the isoform promoted by mutant P53, the levels of active KRAS were actually maintained. However, as soon as we overexpress the minus poly-C GAP17, that isoform that is repressed by mutant P53, we saw that the levels of active KRAS decrease, and also the active levels of ERK, which is downstream of KRAS, were also significantly decreased. And so it was interesting to see kind of like the different effects on the active form of KRAS in the presence or absence of these GAP17 isoforms. So then we went and did a cell-free assay where we took wild-type KRAS or mutant KRAS, and we incubated it with either the poly-C GAP17 or the minus poly-C GAP17. And what you can see is that there was no much difference in the cell-free assays in terms of their capacity to hydrolyze GTP-bound KRAS. And this was very odd and surprising to us, because actually in the cells, they were maintaining different levels of KRAS bound to GTP. So this made us go back to the drawing board and start thinking about what happens in the context of cells in the activity of GAPs. It turns out that GAPs are usually cytoplasmic proteins that when called to deactivate KRAS, they go to the membrane, and that's when they actually promote the hydrolysis of the GTP. What we were seeing was the following. In the presence of mutant P53, when you have the poly-C GAP17 being expressed, the GAP mainly localizes into the cytoplasm of the cell, even when we gave it signals to go to the membrane. However, when you knock down P53 out of the cells, you can see that there is this, the GAP17 that is now not expressing poly-C exon now can more likely reach the membrane and promote the hydrolysis of KRAS. And so we were excited to find these because that led us to a model where we had, for the first time, kind of discovered how these two oncoproteins, mutant KRAS and mutant P53, synergizes in the following way. our model suggests that in the presence of a wild type P53 or the loss of P53, the cells actually lose poly-C exons across multiple mRNAs, mainly the gap mRNAs. And after this mRNA gets translated, it encodes gaps that actually are efficient at reaching the membrane and being more efficient at hydrolyzing GTP-bound KRAS and promoting tumor growth, but not as much as when you have the hotspot mutant, because now this time you are gaining a poly-C exon. And when that mRNA gets translated, it has now this rich domain of prolines that prevent it from reaching the membrane, maintaining an active KRAS state and more tumor growth. So just to go back to our model and the genetics of pancreatic cancer. So I've told you before that you needed KRAS and mutant P53. And what our findings had suggested is that in the presence of just mutant KRAS, you still have the active RAS. But then when mutant P53 comes, specifically the gain of function mutant of P53, you now have an altered RNA splicing and a feedback loop that now prevents the gaps from being active. And then in this way, you can enhance the oncogenic signaling and activity of KRAS. And this is our model system currently. So that was great. And we published this a couple of years ago. So then we came back into the lab and we started thinking, well, how can we target RNA splicing in pancreatic cancer cells? And so recently, a small molecule compound, H3B8800, it started being tested in phase 1 clinical trials, and they got interested in our research with pancreatic cancer and mutant P53. So basically what H3B8800 does, it binds to one of the core splicing proteins, SF3B1, and prevents this whole machinery, the spliceosome, to bind and recognize fully the mRNAs. And so our hypothesis was, well, if mutant P53 tumors really depend on altered RNA splicing, they'd be more sensitive to they would be more sensitive to any perturbation into the splicing machinery with the 8800. So we launched what we call a mouse trial, where we took mice that either had tumors that had mutant P53 in them, so that's red, or that lacked mutant P53 in them, which are blue. And then we randomized these animals to either receive 8800 or vehicle. And what you can appreciate is that the solid lines, which are the animals that receive 8800, they all benefited from receiving the compound. However, the animals that survived and benefited the most were those ones that had mutant P53 in them, suggesting that this mutation sensitizes these tumors to the splicing modulator. And when we did RNA splicing analysis after we treated these tumors with the 8800, we can nicely see how H3B8800 was repressing the retention of that poly-C exon in the cells as a function depending on the concentration. So in other words, this compound was reversing the key splicing events that we had seen in the presence of mutant P53. Lastly, we have now established a human model where we have isogenic cells that express different forms of mutant P53. And we know that when we treat them with this compound, the mutants in particularly are more sensitive to these compounds when you compare them to the counterparts when they don't have P53 or when they have a wild type P53. So based on these results, we are now in discussions with H3B Biomedicine and Roinvad Sciences, who recently bought the 8800 compound, because we would like to start a phase 2 clinical trial where we combine gemabraxine, which is one of the first standard of care chemotherapeutic lines for pancreatic cancer patients, and start escalating doses of 8800 for patients whose tumors have gain of function mutant of P53. So hopefully we can launch this soon. So let's go back to, what are the mutations that drive pancreatic cancer? What have we understood, and how can we target to drive personalized medicine? So as I mentioned before, we now understand that KRAS mutations are the most common mutations, and they are required hit mutation to form tumors. We know that 10% of the cases have familiar pancreatic cancer, and most of them have mutations in ATM. And when they have these mutations, they are given PARP inhibitors. And that's why we do molecular molecular profiling in these tumors to identify this cohort of patients that have actionable mutations. We also know that, as I mentioned before, that 30% of the sporadic pancreatic tumors, which are the most common ones, are driven by gain of function mutant p53. And as I mentioned before, we're hoping to start a clinical trial using 8800, the splicing inhibitors, to see if we can bring a targeted therapy for these sporadic tumors. Now, we're still facing the challenge that we still don't understand what is the mutation that drives the other 30% of pancreatic tumors, because we already know that the other 30% is driven by loss of function p53. So for these two last groups, unfortunately, right now, we don't have any targeted therapy or any trials that are going to launch here. So we were curious to know, well, what other mutations drive sporadic tumors of pancreatic cancer? And so to answer that question, I mentioned before that, there is such a large number of mutations that appear in very low frequencies that it's hard to study each one of these mutations to understand, well, are they driver mutations or are they passenger mutations? And so we took an informed approach where we went back to the basic concept of mutual exclusivity. So just to remind everyone, we know that mutations may be mutually exclusive, meaning that if p53 is present, the mutation in p53 is present, then it would turn into a viable tumor cell. If other mutation is present but not p53, it could be viable, but if both mutations are present, it could be synthetic lethal. And the mutual exclusivity of these mutations is because sometimes these mutual exclusive mutations either have the same function or impact the same pathway, and that's what makes them driver mutations. So we started conducting a mutual exclusivity analysis by taking advantage of cBioPortal, which has the mutation signatures of over 3,000 patient samples of pancreatic cancer. And so the first results that we derive from this analysis are shown here in this volcano plot. So on the right-hand side, we have the mutations that co-occur with mutant p53. And in the left-hand side, we have the mutations that are mutually exclusive for p53. And this was the side of the volcano that we were interested in, because this potentially could tell us what mutations were driving this disease aside from mutant p53. So as a proof of concept, KRAS is in the middle, is the first hit mutation. It appears for all of the tumors, but here's where it got surprising to us. One of the most mutually exclusive mutations to p53 was mutation in SF3B1. It's a core splicing protein. Then on this same side, we have mutations in RBM10, which is another splicing factor. But on the co-occurring side, we had a U2AF1, again, another mutation in another splicing factor. So if our hypothesis was true, it's potentially that mutually exclusive mutations, meaning SF3B1 and RBM10, could be drivers of pancreatic cancer. And just to zoom in on what type of mutations are present in SF3B1 in pancreatic cancer has a driver mutation, a hotspot mutation in K700E. RBM10 is mainly truncating mutations. So basically you're losing the function of RBM10, and U2AF1 has a hotspot mutation in S34F. So our question was, are any of these three mutations driving pancreatic cancer? And so we took advantage and we started generating genetically engineered mouse models. So here's the KC model system where it's only driven by a KRAS mutation. And what we found and what we expected was that these animals should only form pannins or pancreatic intraepithelial neoplasias, those precancer states. So then we crossed this KC animal with a U2AF1 mutant animal for the S34F mutation. And what we found is that actually there are pannins, but not as much as we expected. And most importantly, there was no PDAC in these animals. But surprisingly, the animals that had the KRAS mutation and the SF3B1 mutation formed pancreatic tumors, same as the animals that we crossed to have KRAS and RBM10 loss. So Here's just the quantification done by our pathologist. So you can see that there is only PDAC in the animals that have the mutation in SF3B1 and the mutation in RBM10. There is more pannins also in these animals, and they succumb to the disease very early on. So we are now in this hypothesis that we're trying to further test, which is we believe now that pancreatic cancer cells that have a mutant KRAS actually require a splicing switch in order to become tumor cells. And most likely, the majority of these tumors will develop through a mutant P53, which I showed you before, how it drives alternative RNA splicing. But we're now further studying how these SF3B1 mutation and RBM10 loss also drive the disease based on a splicing change. And these animals are now being characterized by a couple of postdoctoral fellows in my lab. And so I just want to quickly mention that they have obtained really interesting results in terms of what are the splicing defects that these mutated proteins are leading to. They are very similar to the P53 splicing changes. We do tons of deep RNA sequencing into these model systems. We do we run several algorithms to determine the splicing changes into not only the murine model systems, but also patient-derived samples. I just want to skip quickly through this just so I can get here to how are we going to target these mutant splicing factor proteins. So similarly, we use the 8800 compound. And we are now finding that also these mutant cells are very sensitive to this compound. We are also finding that these mutations confer sensitivity to certain chemotherapeutic agents. In this case, in particular, the K700E SF3B1 is more sensitive to gemcitabine than it is to 5-FU. So this is important because these mutation profiling can also help to decide what would be the best chemotherapeutic agent to assign to a particular patient. And when we did combination studies on mixing gemcitabine with 8800 in mutant versus wild type cells, we can see that the mutant cells are more sensitive to the combination of gem and 8800, more so than the wild type cells, suggesting that this combination of therapy could be important to treating the patients that have these K700E mutation in SF3B1. So I just want to finalize by saying that currently, based on our findings on mutant P53 and mutant SF3B1 and RBM10 loss as drivers of pancreatic cancer, all of these mutations leading to changes in alternative splicing, we're hoping to also bring into the trial eligible patients that are K700E mutant or have RBM10 loss to be eligible for this anti-splicing therapy that we want to launch in combination with gem and 8800. And so with that, I want to wrap up by saying thank you to everyone here for your attendance today, to all the people in my lab who are leading this effort, to all our collaborators, and also to our funding sources. Thank you very much. And I'll take any questions. Thank you.
Thank you so much, Luisa. That was really fascinating work. Great, excellent stuff. If people have questions for Luisa, please put them in the chat and I can read them to her and she can go ahead and answer them. I had one quick question while people are formulating their thoughts, which is, it is intriguing that the mutations and effects, and indeed the 8800 are all focusing on U2. Do you have some, is there, it may be my ignorance, but what does that mean mechanistically?
Yeah, thank you, Mark. So basically, the compound targets mutant SF3B1. And so if the tumors have mutant SF3B1, they are more sensitive to this compound. So that's the case for SF3B1. But we also know that if tumors highly depend on splicing, they're more sensitive to this compound because they cannot tolerate a double perturbation of the splicing changes in the splicing machinery. And so that's how we are attributing the sensitivity of mutant P53 and mutant RBM10 to 8800. And I think more dissection of the mechanism of the drug within, RBM10 and P53 can further elucidate why are they so sensitive to this compound. At least for P53, we know that in certain cases, it reverses the effects, the splicing changes that mutant P53 is promoting.
Okay. Makes sense. And the other quick question I had was that with going back to the GAP17 story, do you see in other circumstances if you look through other cells and indeed tumors that don't have the gain of function P53 mutations, do you see the GAP17 with the poly-C exon in other places?
Yeah, so Mark, that's a good question. So for example, we've looked into other cancers that are not KRAS driven, but have this mutant form of P53, and we see that indeed the splicing change in GAP17 occurs. Now, we have also seen some other tumors where mutant P53 is not present, and we still see the splicing change. And we think that this is attributed to the overexpression of a splicing factor called HNRMPK that today I didn't have time to go into, but we think that this splicing regulator also promotes the poly-C the poly-C exon inclusion in mRNA. So we think that there is not a single pathway to promote the poly-C exon retention in GAPs.
Okay. This is fascinating. And I have a question in the chat from Timothy Robinson, who says, "Great talk." I agree, by the way. You described using mutually exclusive analysis to find events within the same pathway. Did you look at GAP17 aberrant splicing based on mRNA directly to identify other drivers?
So I'm not sure if I'm understanding the question. If we looked into other pathways,
I think
that are not linked to the KRAS pathway.
I think the question is actually, did you look at aberrant splicing based on mRNA to identify other drivers that might be other than GAP17? I think that's what the question is.
Yeah, so all the splicing changes that we identified are based on mRNA sequencing and based on splicing analysis that we conduct. But I can also mention that the gaps are only 5% of the events that mutant P53, 5% of the events, the splicing events that mutant P53 is triggering. So there are other mRNAs that affect other pathways that are being impacted by the aberrant splicing by mutant P53. So we just went with the gaps to start with because, of course, of the relevance and the path and the KRAS pathway. But there's a student in the lab who's actually trying to understand what is the role of the other splicing changes in other mRNAs that are not gaps.
And presumably in that context, I mean, it's kind of interesting that the GAP17 effect is so kind of singular in a sense. Presumably in the other cases, it's really going to be a combination. It's going to be the constellation of those changes that are the key, which is going to be interesting but tough to tease out.
Exactly. So as I mentioned before, we are seeing that 32 gaps encoded by the genome of 120 gaps that are encoded are being differentially spliced. We manipulated one, but if you imagine manipulating several of them and forcing poly-C exons to be excluded, the effect might be synergistic in terms of the cell proliferation and the tumor growth.
Any other questions in the chat? So we don't seem to have any at the moment. Anyway, so but we should probably move on. So thank you very much, Luisa. That was fascinating stuff. Enormous amount.
Thank you, everyone.
To think about. Thank you. So let's move on for the second half to Dr. Rosa Munoz Xicola. Dr. Xicola is an assistant professor of medicine and digestive diseases. She received her PhD from the Universitat Autónoma de Barcelona, I apologize for my pronunciation, in oncology, and her postdoctoral training at the Institute of Cancer Research at the University of Illinois and at Yale University. Dr. Xicola's long-term goal is to decipher the unknown genetic alterations that predispose to colorectal cancer development, and her research focus is on understanding molecular and the molecular characterization of sporadic and hereditary colorectal cancer, with an interest in understanding the biological differences among racial groups. To develop her translational research, Dr. Xicola is a key player in several repositories and consortia that recruit cancer patients, and in collecting biospecimens and clinical data. And Dr. Xicola will tell us about defining new pathways in colorectal tumors with mismatch repair deficiency. So thanks so much, Rosa, for doing this. I really look forward to your talk.
Thank you. Let me share. Can you see properly? Yep.
That's perfect. Yep. Great.
Okay. So thank you so much for giving me the opportunity to show you all our most recent data on the topic of Mismadrupper deficient tumors. So the outline of the talk is I'm gonna explain you, give you an overview of the mismatch repair and the phenomenon of microsatellite instability. And then I will explain you the clinical phenotypes and challenges in the molecular diagnosis of the tumors that have mismatch repair deficient. Then I will explain you the association that we are describing between deficiency of RECU DNA helicases in Lynch-like syndrome cases. And then I will show our most recent publication that describes the identification of tumors with a high likelihood of developing an immune response through mutational signature profiling. So here in the left, you can see it's a cartoon that shows the most important or the main proteins that are involved in the mismatch repair system. The mismatch repair system is a DNA repair system that identifies mismatches like single base or like larger mismatches. And there's two main complexes, the MUT-S, that it's formed by MSH6 and MSH2, and MSH3 and MSH2. So these proteins are the first ones to recognize the mismatches, and then the MUT-L complex is recruited to help fix the mismatches, and MUT-L is formed by PMS2 and MLH1. So in the genome, there are these sequences that are called microsatellites that are prone to acquire alterations when any of the proteins of the mismatch repair are not working. So here you can see, sorry, this is on the way. Here you can see a microsatellite. Microsatellites are short and repetitive sequences present in coding and non-coding regions of the genome. And when any of the mismatch repair proteins is not working, these microsatellites accumulate deletions or insertions. So when the size of the microsatellite cannot be properly kept during replication of DNA in the cells, the phenomenon of microsatellite instability is identified in tumors. So MSI can be identified in a variety of tumors, but as you can see here in the table and in the graph, endometrial tumors, colorectal, and stomach are the tumors that have a higher incidence of microsatellite instability. So in colorectal tumors, about 10% of sporadic tumors have mismatch repair deficiency. And this deficiency is due to CpG island promoter methylation of the gene MLH1, which I show you that it's one of the two proteins that forms the MUT-L complex. So when there's promoter methylation, there's an inhibition of transcription of the gene, and it ends resulting in the loss of expression of the protein. So here you can see the difference between normal expression by immunohistochemistry and loss of expression. And a significant number of these tumors, they also present this hotspot mutation in the BRAF gene. Here you have the mutation. And these two molecular events are used to differentiate the tumors that develop through sporadic events than the tumors that develop MSI, but they are developing in the setting of hereditary disease. So Lynch syndrome is the tumor, it's a cancer syndrome that is due to germline mutations in these mismatch repair genes. It's actually the most common cancer syndrome of all. It's present, it's estimated that 1 in 270 people in the US carry one of the mutation in one of these genes. And these individuals present, this syndrome presents as penetrance about 70 to 80%, which means that in 70 to 80% of the cases, individuals that carry a mutation, they end up developing cancer. And when they develop cancer, it's usually associated with an early age of onset. So clinically, Lynch syndrome patients present with fewer polyps than other colorectal cancer inherited syndromes, and the tumors localized on the right side of the colon. And these Lynch patients, they have a high risk of developing multiple cancers, colorectal cancers at diagnosis or over time. And another clinical feature that it's important to remember from these patients is that the Lynch syndrome is actually a multi-cancer syndrome affecting different organs. And here you can see the list. And it's significantly important to remember that because actually female Lynch patients, they develop, for example, like endometrial, they have a higher incidence of developing endometrial than colorectal cancer. So because I explained you that Lynch syndrome is the most common cancer syndrome, and because of all these clinical features that these patients have, nowadays all endometrial and GI cancers are supposed to be tested for Lynch syndrome. So how this works is all these cancers, they are tested with immunohistochemistry for the expression of the 4 main proteins of the mismatch repair. If loss of expression in MSH2, MSH6, or PMS2 is identified, then the patient should be referred to cancer genetics for testing. Contrary, if loss of MLH1 or PMS, or the conjunction of MLH1 and PMS2 is identified by immunohistochemistry, then there is the MLH1 methylation should be tested. And if there is no methylation, then the patient should be referred to cancer genetics. And in any way, if anyone identifies an MSI case, but there was no immunohistochemistry tested, but the physicians have a clinical concern, then this patient should be referred to cancer genetics. So in the cancer genetics clinic, what we've been facing is that about 50% of the suspected Lynch syndrome patients that are referred, they actually test negative for having germline mutations in the Mismadrupper genes. And these cases were named as Lynchlike syndrome because they are similar to Lynchlike, but there's no mutations. So as a definition, these Lynchlike syndrome patients, they develop tumors at MSI. They don't have methylation of MLH1, they don't have the hotspot B600 mutations, and they don't have germline mutations either. So what are these Lynch-like cases? They actually could be Lynch syndrome cases, but that due to difficulty on identifying mutations or because they have like encrypted mutations, maybe we have not been able to identify them. Or they could actually be hereditary cases that they might be due to germline mutations in other genes and that they end up developing MSI as a driver effect, not as a cancer driver effect. Sorry, that they develop because other germline mutations, but they actually, the MSI was an effect of development of cancer, or they could just be sporadic cancers. So to address these challenges, we have developed 2 main projects, one at the germline level and another one at the somatic level. The germline level, our aim was to identify recurrent deficient DNA repair genes and the cellular consequences that contribute to the development of colorectal cancer in Lynch-like patients. And at the somatic level, we aim to define molecular factors in the 3 types of mismatch repair deficient tumors, the Lynch, Lynch-like, and the BRAF methylated ones, which will contribute to diagnosis and treatment. So through our collaborations with the Epicolon Consortium, we were able to describe that Lynch patients have a higher frequency of family history of colorectal cancer than sporadic cases. And you can see here how the standardized incidence ratio was 2.2 for the Lynch in comparison to 0.48 for sporadic individuals. And these incidence of family history was actually lower than Lynch. So this kind of puts the Lynch-like phenotype and in between Lynch and sporadic. We're also able to show that the average age of diagnosis for Lynch-like is significantly younger than in sporadic cases. So these two features suggest that a potential unidentified genetic predisposition induced in a group, at least in a group of Lynch-like syndrome patients. So to address this, and because we believe that that's the case, we develop a study including 654 individuals from our Chicago Colorectal Cancer Consortium cohort, and we performed that Lynch screening testing that I mentioned before, and we identified 23 suspected Lynch syndrome. So from those, we were able to have germline DNA from 15 of them, and we performed exome sequencing, and we identified that 4 of them were actually Lynch, and 11 were Lynch-like, were classified as Lynch-like because we didn't find the mutations. So then we take it one step further, and we wanted to identify if any of these Lynch-like patients had mutations in other DNA repair genes. So we analyzed 162 DNA repair genes, and we were able to see that these Lynch-like patients, they had a higher mutational burden in comparison to Lynch, to the TCGA colorectal cancer cohort, and to controls without cancer. So specifically, we identified 4 loss of function variants, 1 in BARD1, 1 in Werner, 1 in MCPH1, and 1 in REP3L. So then after this first study that we identified that Lynch-like were enriched with mutations in DNA repair genes, we decided to include 2 different independent series of Lynch-like patients to try to identify genes that maybe would be recurrently mutated in this phenotype. So when we did that in the first series, we identified 6 genes that were mutated and had loss of function variants. And interestingly, we found the same splicing variant in 2 different patients in the REQL5 gene. And we actually performed a kinship analysis to show that and to prove that these 2 patients were not genetically, and they were not genetically, they were not related, sorry, because these patients were both coming from Spain, and we just wanted to make sure that there was no any family relation that we didn't know. And then when we developed the analysis of the other series of patients, we again identified another loss of function variant in REQL5. So with that, if you've been able to follow my talk and the identification of mutations in our original series, we have identified 4 different mutations in genes that belong to the REQ DNA helicase family. So here you can see the 5 proteins that are in this family: REQL1, Bloom, Werner, REQL4, and REQL5, and all of them share the same helicase domain. So these are the individuals that we have identified, the 2 individuals with the same splicing, 1 with an insertion, and from the original cohort, we also identified this individual with a Werner mutation. So after that, we were interested in knowing if maybe the mutations in this family of genes were also recurrent in other cancer phenotypes. So to do that, we first took individuals that were referred to the SMILO Cancer Genetics and Prevention Program, that when they were referred and they were tested, in the clinic, they didn't find any mutations in any known cancer predisposition genes. So we performed exome sequencing in 156 breast cancer patients, in 75 individuals that had different types of tumors that were not breast tumors. We also included MSSH and PCC. These are a very rare type of familial colorectal cancer that affects individuals in different generations, and they develop cancer at a young age, but these individuals don't have MSI, they are MSS. And lastly, we also identify mutations in the TCGA. So with this analysis, we were able to see that actually Lynch-like have the higher allele frequency of variants in DNA repair genes that are not the mismatch repair. and that there are not no well-established cancer predisposing genes. And we only identified mutations in the RECQ DNA helicases in the Lynch-like phenotype. So then we went back to the families that we were able to contact again to identify if the mutations were shared, if these mutations were shared with other family members. So here, these are the 3 families that we have with mutations in the RECQL. So family A and B are the ones that share the same splicing variant. So here, this is the proband that developed for family A that developed colorectal cancer at 63. We were also able to sequence the tumor of this individual, and we also found a missense variant in the tumor of this patient. And then the brother of this proband had small bowel cancer, and he was also a carrier of the mutation. In family B, this was the proband that developed colorectal cancer at 64, very strong family history. And then we tested the 2 sons. The one was a carrier and the other one was not a carrier, and their age of diagnosis was on their 40s. And lastly, this last one, this was the proband, developed colorectal cancer at 66, and we tested this son that also had, the mutation. But however, these individuals in the 2nd generation, because they are in their 40s, they might have not been able to develop cancer yet. So this co-segregation study was not definitive. So then we wanted to test what was the effect of having a heterozygous loss of function variant in these genes. So to do that, we went back to our contacts in Spain, and we extracted cells, extracted blood samples from the 2 of these sons that I show you in family B. That one was a carrier and the other one was not. And we extracted RNA, we did retrotranscription and qPCR to show that actually the level of gene expression was significantly lower in the brother that had the mutation. And to test the effect in the Werner, we had to use a different approach because we didn't have access to that family anymore. But we were lucky to acquire lymphoblastoma cell line from a family that had this Werner mutation and heterozygosity, and from a control also. And this mutation is the one that the cell lines have, and it's just like a loss of function mutation just for an amino acids down the line from the actual colorectal cancer mutation that we found in one of the patients. So we grow the cells, we extracted proteins, and we show that, again, that there is an effect on the heterozygous and the protein expression. So with this, we show that when there is when these individuals have a heterozygous loss of function in these genes, they actually have a downregulation of the gene and protein. So then we were interested in knowing, well, if there is a downregulation, what's happening with the activity on the activity of the genes, and how are these cells managing DNA damage? Because again, remember that these are DNA repair genes. So to do that, we grow the cells and we perform a flow cytometry analysis that was actually testing the quantity of phosphorylation of the serine 139 residue of the histone H2AX as an indicator of DNA damage and DNA double-strand breaks. So when we did that, we determined the phosphorylation at different time points, and the black are the wild-type cells, and the gray are the ones with the heterozygous. So it's true that the first time point might be a delay on the phosphorylation. We see that on the other time points there is a higher dose of the phosphorylation, and therefore an indicator that these cells have a higher DNA damage. And here, as you can see, this is the difference between the heterozygous that has like a higher phosphorylation. So, so right now we are also doing more analysis, and we are testing for example, for the effect of these variants in cell cycle, because some of our preliminary data are showing that maybe these cells are actually arrested in G1. but we have not had this data yet. So in conclusion from this aim, we believe that heterozygous loss of function variants in DNA repair genes, such as Werner and RECQL5, could predispose to tumor development because they are enriched among the Lynch-like cancer phenotype, they lead to gene downregulation, and they increase DNA damage. So now turning it to the aim two at the somatic level. To develop this aim, we also included two different independent series of tumors, Mismatch Repair Deficient tumors from the three different types, and we developed exome sequencing to identify somatic variants and loss of heterozygosity events. And with this data, with this exome data, we also were interested in identifying the contribution of mutational signatures to these tumors. So mutational signatures are like a fingerprint it's a portrait of the mutations that the tumor has acquired over the development of the tumor. And they, some of them are well established and they are associated to, for example, exposure to carcinogens, and other ones are associated, like in the case of the mismatch repair, to deficiency on DNA repair pathways. So these are the 6 current well-established signatures that are associated with deficiency of the mismatch repair. So when these tumors have, when the tumors have deficiency and you analyze the mutational signatures, you can see this one. So we were interested in knowing what was the contribution of these signatures to each of the tumors. So let me explain you this colorful graph here. So first we identified what were the mutational signatures that were contributing the most to each of the tumor, and then we performed clustering to see what are the groups of what are the tumors that have a similar contribution of those. So here each row is one tumor, and each column is a mutational, it's a contribution to the mutational signatures. And here we are also having the phenotype. So in here you can see if a tumor is Lynch-like, Lynch, or the MSI BRAF methylated. And then in the last column here, we are showing that which is the protein that each of these tumors have lost of the expression. So when we perform this analysis, you can see we identified 2 of the mutational signatures based on the contribution of SBS 26 and 15, which are very well established mutational signatures associated with deficiency of the mismatch repair. We identified first the 2 clusters that are enriched with the Lynch and the Lynch-like. And then we identified this cluster that has a higher contribution of the tumors that are MSI BRAF mutated. So here I'm showing you the different clinical features associated with each of the clusters. And as I mentioned, cluster 2 is enriched with MSI BRAF mutated. And also this cluster has specific clinical features that are well established with this type of tumors, which are that they develop predominantly in female patients at an older age, and that they are associated with right side location. So as molecularly, as I explained you, this cluster is associated with loss of expression of MUT-L, and mainly due to the methylation of MLH1. And then these tumors, they also have the higher number of frameshift mutations, even though there is no difference in tumor purity that could be affecting this, but we didn't see that there was a significant difference, and they don't have a significant difference either on TMB, so tumor mutational burden. So it's specifically to the frameshifts, and what this suggests is that the tumors in this cluster, they actually have the higher level of microsatellite instability. So one of the results of having a higher level of microsatellite instability could be that these tumors have a different neoantigen load. So neoantigens are these peptides that are generated after somatic mutations arise in the tumor. And as you can see here, you can see that a normal protein, and this is a missense mutation in the tumor. So this is going to be 1 amino acid different from the self, the regular normal protein. But neoantigens that are developed from frameshift mutations are significantly different from the normal, because they introduce a lot of, well, insertions and deletions. So these proteins, these peptides are significantly different from self. And these neoantigens, which is represented here by this dot, are presented through the HLA-1 receptor to the TCR, to the T cell receptors. And this is obviously a very simplified version of what's happening. But then when this is happening, then the T cells identify the tumor cells as non-self, and then they're going to start the immune response. So we wanted to see how these neoantigens and the interaction of the HLA for each of the patients were occurring based on the different clusters that we identified. So to do that, we use several bioinformatic pipelines. We use Polysolver to predict the HLA-1 alleles, that we know that there's 3 of them, using the germline exome sequence data. Then we use Anovar to annotate all the mutations that we had identified in the tumor exome sequencing. And then we use NetMHCPan that actually identifies what are the interactions between the HLAs and the neoantigens. And then we took it one step further and we used NeoPred5, that actually this algorithm computes a recognition potential. So what it does is it provides a likelihood that this interaction is going to occur, and it's based on the immune epitope. This prediction is based on the TCR receptor repertoire that it's present in the immune epitope database. So with that, we took this likelihood and this recognition potential, and we scored them, and we identified the ones that were at the highest 10 percentile and the ones that were at the lower 10 percentile. So we assume that if there's no selection, then these interactions in the top percentile between the neoantigens and the HLA-1 should be, the distribution of these alleles should be similar to the distribution of the patients, the allele frequency in the patient population. So to test this hypothesis, we compare the actual frequency of the alleles in the patient population for each of the different clusters, and the frequency and the distribution of the alleles in the ones that are selected as having the higher likelihood of recognition. And what we identified is that actually there was one specific allele, B702, that was significantly enriched in this in the top 10% recognition potential, which that was not happening in the lower set of interactions. So we think that the specific HLA-1 alleles like the B702 could promote stronger immune response in these tumors that are the ones with a higher microsatellite instability. And we believe that these down the line could be affecting the immune response of these tumors to immune checkpoint inhibitors. So obviously this is the beginning of like expanding this work in the area of immune response, in the immune checkpoint inhibitor response. So in conclusion, for M2, molecular differences between the 3 different types of mismatch repair deficient tumors could have a direct implication on immune response. Specific HLA-1 alleles could be driving the presentation of neoantigens among mismatch repair deficient tumors with the highest level of microsatellite instability. We published recently this work. So overall, the take-home message is that our studies show that there's novel molecular heterogeneity among mismatch repair deficient tumors, and that understanding the clinical pathological features associated with this heterogeneity is essential to accurate diagnosis and prediction of treatment response in the setting of personalized medicine. And our future directions is to understand the molecular mechanisms that associate RECQL5 and Werner deficiency with this type of tumors, identify immune regulators that determine response based on the type, the specific type of Mismatch deficiency, and investigate also the treatment response to immune checkpoint inhibitors based on this type of Mismatch deficiency. So with that, just acknowledge our funding sources, Marcin Kalabuch, that is the postdoc in my lab that has done most of the work, and my collaborators in the US and also in Spain. And I'll be happy to take any questions.
Thank you very much, Rosa. A terrific work. That's really very interesting. And we do have a couple of questions in the chat, so which hopefully I can read properly. So the first is from Jeffrey Townsend, and Jeff asks, is the association of BRAF V600E with MSH mutation purely mutational, or is there some more complex biology to the association? And he asks because the trinucleotide signature induced by MSH is especially likely to make the BRAF V600E mutation.
Yeah, so the BRAF mutation in colon cancer is associated with the serrated pathway. So that's like the more biological, it's not this type of tumors, but for the MSI BRAF, it's more like a mutational association. But the one that has been more described biologically is the one that the serrated pathway, the tumors that develop the serrated pathways are developing. But this is like more like a mutational that we use to really separate the sporadic from the hereditary ones.
Okay. Great, thanks. And then the next question is from Ryan Jensen, and Ryan asks, one of the potential roles of RECQL5 is to prevent aberrant homologous recombination by displacing RAD51 off single-stranded DNA. And he wonders if in tumors from patients with loss of function mutations in RECQ, do you see increased chromosomal aberrations, sister chromatid exchanges, or perhaps increases in microsatellite contraction or expansion?
So all of these so the work done in RECQL5 and colorectal cancer is not very vast. So right now, what I can say is that we just engineer a cell line that has these mutations through CRISPR. So we are going to have the cell lines that have like the heterozygous and homozygous and wild type. So we are going to be testing these kind of events that Ryan is suggesting. So I don't have that information yet. What I know that, for example, for RECQL5 is that there was one old paper that was showing that, interestingly, RECQL5 downregulation was identified in MSI tumors. So I think that there is more that we can be learning about this, and I think that that's going to be one of our next steps.
Great, thank you. And I had one quick question. When you were going through and looking at the RECQ and other mutations in the Lynch-like syndrome, I didn't have a sense for whether it was clear that they're all loss of function mutations, like for example the T31K in that one family. Is that a
no, that one. So that one was a mutation that we found in the tumor of that patient. We have not been able to test the other individuals. So the germline variants that we are identifying in the germline are all loss of function, but we only have been able to test one tumor from these individuals. I can tell you, not for RECQL, I know a lot of the data for, well, not a lot, few data from Werner mutation somatically. They're identifying loss of function mutations, and actually these tumors that have loss of function mutations in Werner, they have a significantly higher number of them in comparison to the ones that don't have mutations. They are MSI. So again, another kind of another clue that there might be some association between deficiency in these genes and MSI. However, association doesn't mean causality. So this is what I think that is what we actually need to do more research to figure out if it's one or the other.
Cool. Good. Well, I think it's been a great session, and lots of good questions and two fantastic talks. So I'd like to just to finish by thanking Luisa and Rosa very much for really giving very stimulating and exciting talks. Great grand rounds, and thank you very much, everybody.
Thank you.
Bye-bye.
Bye.